I can raise us a bit more if you want.
No.
Yeah, that's inspired.
Okay.
Good afternoon. Those who don't know, my name is Jonathan McKee. I'm the college president. So welcome everybody to Keller College. Welcome to everyone who's watching around the world. I should say that there will be family and questions and discussions. There are government questions as you don't go through. Do keep them to the end. And then we're looking online. If you put your questions into the chat, then we'll have your questions in it as well. This is an annual McLaren lecture. I should declare interest, and McLaren was my daughter Lee and Caroline here, who's a geneticist. I won't say more because you're going to hear more. But at the event this time last year, both Professor Sarah Franklin and Professor Emily Jackson were on the panel talking about various aspects of their research and Anne's work, and mentioned that they were working together at a time on this book, which is literally hot off the press on the 14-day rule. So I asked at the time, would they come back in a year's time to, in effect, do a book launch? And I'm pleased to say we've got some copies of the book at the back, hot off the press, which the authors have very kindly agreed to sign. They've already signed the copies to Keller College, but they'll sign them for you too. So, can I invite you both up?
Certainly.
To,
Yeah.
the presentation, and then we'll do questions and answers.
Yeah. Thank you. Yeah. So thank you very much, Jonathan. It's great to be back here again. I'm very pleased to be here to launch our book, which we've really enjoyed writing together, and which is dedicated to Anne McLaren and to Mary Warnock. Partly, I think one of the reasons we wanted to do this book is because we felt that the legacy that Anne McLaren and Mary Warnock have left in policy terms, in terms of creating effective science policy, the story of how they did that, why they did it, the way they did it, and so forth, hasn't really entirely been told. So this book makes, I think, at least a start in telling that story.
Right, shall I put the next slide?
Yeah.
So yeah, so what it does is it really talks about what the 14-day rule is. Just to recap for anybody who isn't aware, this is the legal limit on which you can culture embryos. Sorry, which you can culture embryos carrying out. And we think that this rule is actually, and its development is pretty striking, not just because it has held as a limit in this country for decades now, but it's been copied worldwide. But I think what we're really interested in is how it came about and the role that Anne McLaren played in that.
Yeah.
Next slide.
Yeah. So the book is divided into chapters that tell the history of the 14-day rule, how it came about scientifically, why was the research being done around embryos to begin with, and then was the legislative history of the rule, which is quite a sort of lengthy period of time over which it was developed. Then what has been the sort of lifetime of the 14-day rule, what's happened since it was passed in 1990, and what's the future of the 14-day rule, because recently there have been calls to change it, which we'll talk a little bit about today. And then really the most important part is, what are the lessons to be learned from this particular policy chapter? So just to start sort of at the beginning, as it were, many people will be aware that the birth of Louise Brown was very significant, both clinically and scientifically, but of course it was also very significant legally, because with the birth of Louise Brown was seen also to have been born a legal vacuum. it immediately became clear that there was no legal framework for regulating either in vitro fertilization or the research on which it depended, embryo research. So in 1982, a committee of inquiry was established. It was initially called the test tube inquiry, the test tube baby inquiry. There were 16 members of the Warnock committee. The chair, Mary Warnock, was a philosopher. Most of the people on the committee were not scientists. So it was a bit of an unusual committee in that it marked a transition away from the idea of scientists making decisions about what scientists do to much more of a lay public committee that would debate all of the different aspects of what came to be known as human fertilization and embryology. So it's worth saying that Britain is the only country that has ever successfully regulated in this area. It's a bit surprising, but actually no other country has ever successfully passed comprehensive legislation around IVF and embryo research. So the way in which this took place is unique, which is another reason why it's quite an important story to tell in more depth. Yeah, go ahead. But until recently, it was quite difficult to really give a detailed account of how the Warnock Committee did its work because the files were embargoed after the committee ceased its work, after it published its report in 1984. And nobody really knew where the files were once they were embargoed, because they were embargoed for such a long time. So it was really actually purely by coincidence that members of my research group found a nearly complete set of the Warnock files in, of all places, Burnley, Lancashire, in a Department of Health repository, which we didn't even know existed until we asked to look at some of their more recent files, which were the only ones we could find related to the Warnock Committee. And while we were there, one of the librarians said, "Oh, would you like to look at some of these?" And they were preparing them for the National Archives. So yeah, go ahead. So just to go very quickly to the kinds of things the files have enabled us to understand about the committee and its work. It was quite clear very early on that the Warnock Committee would face a great deal of opposition to IVF and embryo research. People in general seem to be okay with the idea of using IVF to help infertile couples have babies, but they weren't so happy about the idea of embryo research, human embryo research on embryos that were being created in vitro in the lab. And so there was clearly going to be quite a bit of opposition. And, you know, people don't always remember either that when Louise Brown was born in 1978, it was right before the election of Margaret Thatcher. And with the election of Margaret Thatcher in the following year, 1979, there was a huge shift in what you might call political values around the family, reproduction, sexuality. And Thatcher was quite famously associated with a turn to a more, Victorian view of the family and gender and kinship and reproduction. And so it very quickly became apparent that the anti-abortion movement would be very opposed to IVF and embryo research, and indeed they were. So this document, which you can't really read, I'm sorry, it's not high enough quality, but this document is the very first item for discussion on the 9th of November, 1983, when the committee met unusually to discuss a single issue, which was the question of whether human embryo research should be allowed at all, and if so, how. And this was the very first document they were given, which is an analysis of public correspondence on the matter. And this tabulates all the letters they've received in favor of IVF, letters written in opposition to IVF, letters written in opposition to embryo research, and letters written in opposition to both. And what the numbers showed was that 50 times as many members of the public had written in opposition to both IVF and embryo research than had expressed any positive support for it. So the very first document the committee was given on 9th of November 1983 was a document that showed 50 times as many people opposing these IVF and embryo research than in support of it, which clearly was partly the result of the campaigning around the pro-life movement, which of course is a very famously well-organized campaign, also supported by a number of very prominent members of parliament, but still a very daunting way to start. Yes, so next one. So the other thing that becomes very clear from the papers that we found, from the documents in the Warnock files, is what Mary Warnock's philosophy was towards dealing with the embryo research question. And she has written quite a bit about that. So we do know quite a bit about her sort of guiding philosophy of this. And she was very clear that no law would ever successfully be based on a moral decision about the ethical status of the human embryo. She argued that it would never be possible to know absolutely what is the right way to treat human embryos. And she argued that what was needed instead was a means of determining what would be all right to enough people to enable some legislation rather than none. Do you want to do the next one?
Yeah, sure. So I think what's clear from the Warnock papers is this idea that compromise is essential. You're never going to get consensus on something as polarizing as the moral status of the embryo. So Mary Warnock with McLaren Lecture really cut through that and just said, well, we can all agree that some legislation is better than none. So I think what we've got here is the principles that she applied to this work. So people feel strongly about human embryos, and we need to respect that. That's important, how people feel. We're never going to agree on exactly what the moral status of the embryos or what the law should be. So we need to come up with something that is okay. It's not going to please everybody, but it's going to ensure that there is some legislation in place. And this was, I think, a really groundbreaking approach to scientific innovation. It wasn't trying to say, I found the right answer. It was saying there isn't a right answer. We've got to try to find some way through this disagreement. And part of this, I think, and I think something that Sarah and I feel about really strongly about, is that Anne McLaren and Mary Warnock were real pioneers in the importance of hearing from the public and hearing what the public thinks about embryo research. And Anne, I think, in particular, had tremendous confidence that if people have facts explained to them really clearly, they give them information in a way they can understand, they will come up with sensible suggestions. She was a really great believer, wasn't she, in the importance of taking seriously what the public think. And we particularly love this picture of Anne with a mouse.
Yeah. I don't know if you can see that she has very cleverly put the mouse wheel inside the lid of her typewriter, which is one of the many things she did in her lab, which was obviously a space that she really enjoyed being in, where she invented a lot of the techniques that she used, and where I believe her children were often invited to accompany her as well, which may or may not have involved watching the mice run around in the wheel in her typewriter.
So the other thing that this is also, I think, another really groundbreaking thing about the way in which Mary Warnock approached her work is she again recognised, and this was really unusual for the early 1980s, that legislation, regulation is actually good for scientists. It's not just good for the public, it's not just good for society, but actually it's helpful for scientists. So that the scientists, if they know where the boundaries lie, they can just get on with their work, knowing that those boundaries have been set. If you've got to worry all the time about, is this okay? Am I allowed to do this? Actually, it doesn't free you up to do your work. So I think another really groundbreaking element of the Warnock Committee is the idea that regulation is a facilitator of science rather than an inhibitor. And also that it's good for Parliament rather than scientists to have the burden of deciding what those bits be.
So our book is somewhat perhaps teasingly subtitled A Sociology of Biological Translation. So that's kind of to get people to read the book to work out what that means. But biological translation is the way in which scientific ideas are translated into applications. So for example, IVF is considered a translational technology because it successfully took a basic scientific principle about how you could create an embryo, and then it turned that into a successful clinical technique. In order for scientific translation to happen, it's not enough just to have the scientific idea or even to turn it into a particularly effective clinical application. You also have to have social acceptance. You know, you can have the best vaccine in the world, but if people don't accept it, if they don't understand it, if they don't trust it, they're not going to use it. So, so translation requires, we argue in the book, good biological translation requires a sociological understanding of technology as well as a scientific one. Which of course isn't to say that a sociological understanding isn't a scientific understanding, but that's another matter. So what we argue in the book is that one of the reasons that Anne McLaren and Mary Warnock were so successful in creating legislation that is not only the only successful legislation ever passed anywhere in the world about these issues, but is legislation that is still successful so long afterwards, it's still in place, is that they had a very sociological understanding, and that they basically put forward a proposal for legislation that offered a kind of social contract, a very specific kind of social contract. And what it said was that essentially in exchange for allowing IVF and embryo research to take place, these techniques would be subject to the very highest level of scrutiny and backed up by criminal law. They also put this as a kind of reciprocal equation, in the sense that if there were good laws regulating the science, people would be more likely to benefit. The returns on that research would be more widespread, and the research would be improved by a more favorable context, by greater public acceptance of these techniques. So yeah, so we use this expression, a sociology of biological translation, to express that idea.
And also really critical here, and this particular quote from the Warnock Committee, I can almost hear Mary Warnock saying the last bit in her very brisk headmistress-y way, and she was also a headmistress as well as being
Handy.
all of the other amazing things she was. And that was just the idea that the key thing here was to set some red lines, just to find some limits that could sort of embody the social contract by which scientists were going to be allowed to do something that, as Sarah mentioned, the vast majority of the public appeared to be against at the outset, but in return for allowing them to do it, there would be some really hard and fast limits, because a society, very society, depends on the existence of those limits. And so this is, I think, this quote from Warnock also is, I think, for me, absolutely critical, which is just a really deft sidestepping of an intractable debate to say, look, there aren't questions of fact here. There aren't straightforward answers. So let's not get bogged down in those massive philosophical questions about the moral status of the embryo, on which pages and pages and whole libraries have been written. Let's cut to the really practical question. How is it right to treat the embryo? So let's focus not on the big moral questions, but the question of how the embryo should be treated in law. Do you want to?
Yeah, so they basically said No one is ever going to come to agreement about when human life begins. It's impossible. And if that is the question that's being asked, there won't be any successful legislation. And there isn't in most countries in the world for that very reason. In the United States, in Germany, in Australia, and Canada, even in Canada, there's no legislation. None of the Nordic countries, none of the European countries, none of the Asian countries have any comprehensive legislation in this area, although almost all of them do follow the 14-day rule in principle, even though it's not legally binding. So this took a long time. It took a very long time for Warnock and McLaren to bring this very innovative approach, this very innovative, really essentially sociological approach to developing legislation through to fruition. They formed as a committee in 1982. They published their report in 1984. A member of parliament attempted to ban IVF and embryo research in 1985. In 1986, sort of initial kind of draft legislation was created and debated, and it wasn't until 1988 that the bill was actually introduced into parliament, and it was debated throughout the 1988-1989 parliamentary session, and the Human Fertilisation and Embryology bill was eventually passed in 1990, putting into law, enacting all 64 of the original recommendations of the Warnock report, which is also a really remarkable achievement. But yeah, so it took a very long time to put in place this very unusual policy framework.
And during that time, I think what was really key is that McLaren and Mary Warnock didn't just think we've written our report, we're just going to get on with the next thing in our lives. They campaigned really vigorously for this and did a lot of public outreach, public engagement on this. So as I've mentioned before, public engagement was absolutely central. And I think for Anne particularly, I love this quote from this interview she did it with Jane Denton: the more consultation, the better. But also, she didn't see it as top-down public education where you tell the public what to think, because actually the public understand more than they're given credit for. Public engagement is absolutely central. I think as part of that, it's really important that information that scientists give to the public doesn't overpromise and make wild claims about what's going to be possible imminently, but also important it doesn't underpromise by saying some scary thing couldn't possibly happen when we might not know whether that's the case. Yeah. So I think what we think there is what Sarah and others have described as the Warnock consensus. So you're saying that.
Yeah, so 35 years after the Warnock report was published, and more than 20 years after it was passed, two political scientists, Sheila Jasanoff and Ingrid Metzler, wrote an article in 2019 in which they described what they called the benefits of the Warnock consensus. And it's, worth looking at this quotation where they point out that in no other country have very innovative and often radical biomedical innovations been welcomed by the public with a minimum of opposition, not because the innovations have been introduced without public debate, but because they have involved so much public debate. So they use this term, Sheila Jasanoff, very eminent political scientist at Harvard, and Ingrid Metzler, a German sociologist. They argue that the Warnock consensus has really been a hugely beneficial legacy of the Human Fertilisation and Embryology Act.
Yeah, so the science and the clinical treatments that result from it have by no means stood still since 1980s. There's been a huge number of some really very dramatic changes. But because what a previous chair of the HFEA, Susie Leather, described as a quiet environment for this work in the UK, people understand that it's slightly regulated and there isn't that much public debate or challenge to it. It's enabled actually some quite dramatic scientific developments to be accommodated within a framework of tough regulation. And this, I think one of Sarah's favourite pictures is probably one of them.
Yes, I have written another book about Dolly the sheep called Dolly Mixtures. Yeah, I mean, you'll remember this when Dolly the sheep was cloned, and the main question people asked was, well, should it be allowed to clone humans? But actually, there's a lot of other questions to ask about Dolly the sheep, like why was she cloned? Why was she a sheep? Was she really a clone? What was Ian Wilmot's intention? And what Ian Wilmot did when he created Dolly the sheep was he did a very complex series of technological innovations that required not only the transfer of the nucleus from one cell into another, but control of the cell cycle so that that cell could activate and gestate and produce a live, viable offspring who was herself capable also of having of offspring herself. So Dolly the sheep really represented quite a radical transformation in understandings of biological reproduction. And although the millennial period when Dolly and the Human Genome Project were happening are often associated with the completion of the Human Genome Project, which was supposed to revolutionize human medicine and so forth. It didn't. Actually, almost all of the really significant biomedical developments since the 1980s have come from developmental biology and reproductive biology, not from the Human Genome Project. But yeah, maybe, because she was a sheep, and sheep are very good at IVF. And maybe because Ian Wilmot, looks a little sheepish. This was a very interesting episode in what we might call public understandings of science, because there was a very sort of double-edged relationship to Dolly that on the one hand, it's a clone, but on the other hand, really sort of almost a sort of friendly face of science. Right, so this, I just put this in to very briefly mention that when we think about the legacy of IVF, when we think about the legacy of the Warnock report, when we think of, the policies that have been made around human fertilization and embryology, we're talking about a very significant kind of cultural change that in some ways has really transformed ideas about reproduction and biology. And this image, which is an image of microinjection, has, I don't know if you recognize this, has become a sort of visual shorthand for the whole field of reproductive biology. So when there's an evening news item about some new technique, they often use this image of microinjection, even if it has nothing to do with microinjection often. And this image has become almost a symbol of the changes in how reproduction has understood. And I think it's an interesting image because it's not, I don't think, generally received as a kind of Franken image, as a kind of threatening image. But I'd be interested to know if you have thoughts on what this image says or represents. Yeah, go ahead. Yeah.
So we're going to talk now about one aspect of the future of the 14-day rule, and that's regulation in this area, and that's the really critical question of how well-established system of regulation might apply to embryo models. And this is one of the biggest, most interesting questions facing the law in this area at the moment. And we also, the other thing that's really critical here is that it has become possible in recent years to culture embryos for more than 14 days. That wasn't possible at first, which makes it in itself an interesting limit because it was a hard and fast line, but one that scientists couldn't reach. So the combination of being able to keep embryos in vitro for longer and the development of embryo models raises some really interesting questions, we think, for the future of the 14-day limit and future regulation of embryo research. So should I say a bit about embryo models now?
Yes, great.
Okay, so you may have seen coverage of this. There were a couple of really groundbreaking announcements last year. I'm sure there'll be more to come. And this is extraordinary new science where scientists are making model embryos out of stem cells. And they're sometimes described as synthetic human embryos, but scientists don't like the word synthetic because these are very much human. They're made from human stem cells, often derived from a skin cell taken from your tissue donor. So you derive a stem cell line, and you can use that to make these stem cell models. They're really quite extraordinary. that is a picture of embryo model from Naomi Morris's lab at the Crick. So why are scientists wanting to do research on embryo models? Well, it's not just to test what you can do with stem cells, so that's undoubtedly interesting. The key thing, I think this is where it has potential to be really transformative, is that they allow us to understand much more about embryonic development. There's so much we don't understand about the early development of an embryo. There's so much we don't understand about why IVF fails so frequently. We don't understand why miscarriage is common. We don't understand causes of stillbirth, and we don't understand the sort of congenital abnormalities that lead to childhood mortality as well. So there's a lot we don't understand, in particular in what's described as a black box of 14 to 28 days. When it's illegal to carry out research on embryos, and there simply aren't any other sources of embryos. Later in development, women who are having terminations of pregnancy donate fetal tissue for research, but we don't understand much about this. This really matters because it's in the 14 to 28 day period that gastrulation happens, when organs, the very first beginnings of organs start to happen. So obviously it's a critical moment. So how does the law apply here? Well, very briefly, this is the law that Mary Warnock and Anne McLaren devised, and it's these strict restrictions as part of the social contract that Sarah mentioned. You can't do research on embryos without a license from the HFEA. You can't do research beyond 14 days or the emergence of a primitive streak, which happens around there. You have to be carrying out research for one of the statutory purposes, so you can't decide to do it for any reason you like. It has to be things like development of better fertility treatments. You have to prove to the satisfaction of the authority, HFEA, that it's necessary to use human embryos, that you couldn't do it any other way. And as Sarah mentioned, this legislation is backed up by really strict sanctions. If you culture an embryo in vitro for more than 14 days, you can go to prison for 10 years. Now, that might seem really quite dramatic compared with sentences that we hear for some other offences, and to my knowledge nobody ever has, but still it's a hard line with a hard penalty. So do these restrictions apply to embryo models? Well, almost certainly not. The legislation doesn't actually contain a definition of the word embryo. Section 1 just says, in this act, embryo means a live human embryo. As should be obvious, that would be a very weird definition because it contains the word it's defining. It's a bit like saying a table is a round wooden table. It tells you what type of table you're thinking of, but it doesn't tell you what a table is. So the House of Lords, which used to be the highest court in the country, said, and I think they're obviously right about this, this assumes that the reader knows what an embryo is. It's not a definition. So what that means is that embryo has its ordinary language meaning for the purpose of the law. And scientists and the HFEA don't think embryo models are embryos. So at the moment, they're not embryos, they're not covered. So it would be possible to correct that and bring these embryo models within regulation. But the only mechanism the Act gives you to do that is a real sledgehammer. And that's to say they're exactly the same. The Secretary of State can have regulations that said embryos include other things. That would halt all of the research going on at the moment, and most of it wouldn't be able to continue. So bringing them within legislation, within regulation, would be a real sledgehammer. So at the moment, what's really interesting about embryo models is that they are pretty much unregulated. Now, some people have suggested that embryo models could replace embryos for the purposes of research. So I put an acronym in there, I should have got rid of that. I think that's not the case, that it's not going to completely replace it for a number of reasons. One is that in order to check that models are developing in the same way or similar things happening, you need to validate them. So you need to compare them with embryos. And that actually is giving real impetus to extending the 14-day limit so you can validate at the later stage. But some people have nevertheless said we should be trying to replace the use of human embryos in research, a bit like the 3 R's approach that applies to animal research: to replace, reduce and refine. And currently, the law does contain a statutory presumption that you use embryo models if you can to do research, because you have to prove that it would be necessary to use embryos, that you couldn't do the research on models. But I think rather than thinking of embryo models as a replacement, I think we should also think seriously about quite how much patients undergoing IVF often positively want to donate leftover embryos to research. Sarah and I were both at a conference last year where a scientist at Cambridge, Kathy Nykan, explained the lengths some patients go to, ringing up research labs saying, I've got embryos I'm not going to use, please can you tell me how they can be used? Because people, I think, feel very strongly that their children wouldn't exist without this research. And so the desire to give something back is powerful. So I think the idea that we should replace embryo research, as well as probably not being scientifically sensible, I think we also need to respect patients' wishes here too. The key difference between embryo models and embryos is scale. One scientist said to me recently, you could make millions of embryo models really quite quickly, whereas obviously embryos are a scarce and precious resource. What that facilitates is toxicology testing, which obviously would be illegal as well as impossible on embryos. And that could be really important because there is so much we don't understand about how medicines affect developing fetuses. Most pregnant women who need medication are engaging in a game of almost Russian roulette. They're either taking medication which hasn't been properly tested, or they're giving up medication that they might need. If we think about what the moral status of an embryo model is, I don't know whether you want to say anything about potential. I realise I'm going on a bit here.
That's good. Well, I mean, it's a very understandable question, and it's often the most common question that's asked about this area. And as Emily says, the lack of regulation has created a huge debate about what to do about embryo models, which, like IVF, are currently, when it first came out, are currently in a legal vacuum. But when people say, you know, what's the moral status of the human embryo, of the embryo model, what they are often trying to think about is, you know, should there be some sort of qualitative, some sort of quality of the embryo that should determine, you know, the way it's treated morally? Like, should the potential of the embryo, should the source of tissue that made the embryo, should the fact that the embryo model is, you know, human, what kind of human? These questions are often asked, but of course, what we know from the history of regulating embryo research before is that it's extremely difficult to answer those questions, not least, as Emily said, because embryo models come in such a vastly diverse range of forms.
And in relation to embryo models, their potential is one of the big unknowns. We simply don't know what their potential is. The question of whether they could implant is a question to which the answer is both unknown and unknowable, because the experiment you would need to do that to answer that question would be incredibly dangerous and unethical. So nobody in the UK is wanting to put embryo models into uteruses. That's absolutely not what people want.
What they're for, yeah.
So at the moment, we just don't know. There's a lot we don't know here. So just running through questions about how this research should be regulated in the future. We'll need to identify a regulator, and it seems obvious that the HFEA is the appropriate regulator here. It's got more than 30 years experience, and it's dealt with lots of innovative new things in the meantime. One interesting thing about not implanting these models in uteruses is, as though it would be totally unethical, it's not 100% clear that it would be illegal, because the Act has a slightly unfortunate form of words. It tries to prevent this. It says no person should place in a woman an embryo other than a permitted embryo. You can see the obvious problem here. If these things are not embryos, then that prohibition doesn't apply, because this is just prohibiting putting back an embryo other than a permitted embryo. So that, I think, is a loophole that nobody wants to exploit here, but it certainly should be plugged, and it should be illegal to put these things back into a woman. Also illegal to put these things back into an animal. One of the questions with which scientists are grappling, and social scientists and regulators and policymakers grappling with at the moment, is what could replace the 14-day limit for embryo models. And this is really difficult because embryo models, Naomi at the Crick explained to me that it's a bit like Lego. They're modular. You can make them in different ways. They don't have a day one, and they don't develop in a day one, day two, day three, day four. You could jump an embryo model so it's modeling something at a later stage of development. So the time limit won't work. So one of the things that's really exercising the scientific community here is, what should that limit be? If the time limit doesn't work, what other limit could you come up with? Well, the International Society for Stem Cell Research has said you keep them for the minimum time necessary to do the research. Now that might be sensible, but you can see how that's infinitely expandable as your objectives change. So that certainly doesn't have the kind of teeth that the 14-day rule had. As I think Mary Warnock once said, everybody can count to 14. It's easy, you can just count. What is it we could put in place for embryo models that would serve a similar purpose? And I think at the moment we just don't know the answer to that. I think another really interesting question is about consent, because to do this research, scientists are getting induced, they're using induced pluripotent stem cells with stem cell lines from biobanks. Would consent to making embryo models need to be specific? Should you know, if you were a tissue donor for stem cell research, should you actually be giving specific consent to creating embryo models, or is generic consent enough? And then there's an interesting question about, should the penalties be as tough as they were for embryos? Do we need criminal penalties? Well, possibly we do for putting these things back into a uterus. So clearly, as part of the point of thinking about how embryo models should be regulated, key thing is we need public dialogue.
Yeah, so fortunately, some lessons have been learned, and right away, both the UK government and the International Stem Cell Society recommended that public engagement be an absolutely crucial part of answering these incredibly difficult questions, which, as Emily has just explained, really have absolutely no clear points of reference even. So this dialogue project was initiated very recently, last autumn. The Human Developmental Biology Initiative is a research program at Cambridge. They worked with ScienceWise, which is one of the big public consultation bodies here in the UK, and they conducted a very interesting public engagement exercise that involved getting 70 members of the public to agree over a 2-week period to attend a series of online and in-person workshops, where initially they were, introduced to what is an embryo model by scientists, by clinicians. What is it? What would it be used for? What are the legal options, etc. And they were invited to share their views. And the findings were very, very interesting from this. I'm sure there's going to be a lot more public engagement, and there's going to have to be a lot more public engagement if there's going to be a good legislative infrastructure for this research, and also if there's going to be a continuing consensus about what kind of research is acceptable, what kind of research is appropriate, what kind of limits should there be. So yeah, a lot of people were very struck by the lack of any legal guidelines. Obviously it's a concern, and people felt equally that there should be, it is absolutely vital that there should be clear guidance around the use and creation and misuse of embryo models. And although at the moment there's an essentially voluntary kind of self-regulation within the scientific community, that's not necessarily, to many people, the most persuasive long-term option. So clearly there will have to be legislation, but people can appreciate, how difficult that would be. So one of the interesting findings of this public engagement exercise was that a strong recommendation was that there should be much, much more public engagement and much more dialogue around what kind of governance process should happen. There were also some specific findings that were quite interesting. I don't know if you can, maybe it's a bit too small to read. A lot of people hadn't actually heard of the HFEA, but when they learned about, the more they learned about the governance process here in the UK, the more confidence they had in the research. So that basic principle that public dialogue can strengthen trust and openness to more radical scientific research was very much confirmed by this exercise. There was also a lot of respect for science and scientists, which again suggests the the idea that the Warnock consensus has over time built a stronger confidence in radical bio-innovative science here in the UK remains the case. There was, as I said, a desire for a lot more dialogue with scientists, and participants suggested things like making dialogue with scientists working in these areas part of the national curriculum, making it much more of a media event. There was a very, very strong desire for more dialogue around science that came out of this. And another of the very interesting findings was that one of the main reasons 70% of the participants thought that embryo models should be developed in the future was to improve IVF. So there's been almost a little bit of a sort of circular effect here, whereby the legalization of IVF, its development, particularly here in the UK, has made people aware of its benefits, but also of its failures. And the concern about IVF failure was one of the main reasons people thought embryo models should be developed. So addressing the governance gap going forward will undoubtedly be one that is going to involve a lot more discussion and debate. And hopefully this is an introduction to you of what these issues are going to be. This debate probably won't take as long as the one previously about embryo research in the 1980s, but hopefully that's because some of the lessons have been learned and can be implemented about how best to reach a consensus about what should happen.
Yes, and finally, as well as public engagement, there's also just public familiarity changes people's attitudes towards what initially seems really scary. And I really like this quote from Deborah Spar. It's in relation to something slightly different, but she talks about the history of assisted reproduction. Is that we create a new technology, and initially we think it's a bit scary. We're worried about mucking about with nature, designer babies, or madmen hatching Frankenstein. But then she says we discover it's just a nice couple next door, and that becomes far, far less scary. So I think public engagement, public familiarity has really transformed IVF from test tube babies to something that all of us know about in our day-to-day life. And I think that's a history to which we owe Anne McLaren and Mary Warnock a huge debt, because I think the status of IVF and embryo research in this country is down to those two remarkable women. And we'll finish it there.
Thank you very much. And we're happy to take questions if there's time.
Sit down there. Then.
Sure.
Okay, well, thank you very much. It's incredibly interesting and educational for me. We have got lots of stories about Anne and her mice. I'll maybe just tell one, which explains really why Anne got to be on the Warnock Commission and why we spent so much time and effort trying to get the regulation in place and so on. Which is she worked with mice. She was the first person in the world to perform an IVF on mice before they then use it on humans. And she was asked once whether she wanted to be made a dame, and said no thanks, wasn't really her thing. But later on she was made foreign secretary of the Royal Society, and it was the first time in their 300-year history they'd ever had a woman officer. And so they asked her again to be made a dame, and she honestly just didn't want to create a fuss of the newspaper saying, you know, mad scientist, officer, queen, whatever. So she agreed to be made a dame, had to go on to Buckingham Palace, and the queen said, "What do you do?" And saying, "Well, I'm a leading stem cell scientist," which was true, or "I performed the world's first IVF birth," or "I'm the first ever woman officer of the Royal Society." She said, "I work with mice." How interesting. Now, coming on to questions, the details of the research in the book are on the fly you've got there, as well as some details about the authors. But I should have said it may help you in formulating questions and knowing who might be more appropriate for each. Is that Sarah Franklin is the Professor of Sociology at the University of Cambridge and created the Reproductive Sociology Research Group, which she now leads. And Emily Jackson is Professor of Law at the London School of Economics and was actually Deputy Chair of the Human Fertilisation and Embryology Authority, which is set up to regulate the field. I'll open up to the questions to people watching live as well as here. But can I just ask one myself first, because I wasn't aware until you said it of the fact that we were literally the only country in the world which had managed to do that. So what I was wondering is what the state of play is in other countries. How do they manage the situation not having clear legislation? Do the regulators just have to sort of play it by ear?
Yeah, that's a really good question. Do you want to say say something about that?
Yeah, so I mean, obviously it depends. It varies from country to country. I mean, there are countries that have prohibitions in place. So some countries might say we're not going to allow egg donation, or we're not going to allow surrogacy. So there are countries which have laws. It wouldn't be true to say there are no laws that touch on any of this area. But I think it's the comprehensiveness of the regulation that we have, and the fact that we, and again, this was such a good idea in 1984, so groundbreaking, the idea of regulating with a regulator that grants licenses, which can then be quite flexible and respond to changes, respond to new developments. So I think one of the great successes that the HFEA had in the past, which you can't use regulation, you can't use law for, because law moves so slowly, is using a code of practice and license conditions to drive down multiple births from IVF, which used to be over a quarter of all IVF births were multiples, with obvious health risks for babies and for women. And by a combination of the kind of licensing powers that it had, it was able to drive that down to, I think it's somewhere around 5%, 6% now. So it's the setting up of a comprehensive regulator and comprehensive regulation which covers everything that no other country has successfully done.
Right, thanks. Good. Yeah.
It's a very fascinating talk. And I was very interested in the embryo models you were talking about in relation to toxicology. And I just wondered to what extent that would allow you to do testing across a wide genetic basis rather than is possible at the moment.
Yes, I mean, it's an incredibly important aspect of this research, because it has the potential to radically improve the testing of medicines, because as we all know, lots of medicines are initially tested on animals, and animals are very poor models for testing medicines for human consumption. So that's why we got thalidomide. It had been tested on animals, but just the animals that had been tested on didn't show that it was going to have that effect in humans. So the possibility of replacing animal testing with embryo models is really dramatic. And I think people feel very optimistic about it. It means that there will probably, when you first start clinical trials in actual full human beings, you'd have more knowledge that it was likely to be safe and work. So there are all sorts of stages of the drug development pathway that this could be really useful for.
Yeah. I think the whole area has expanded so quickly that the options for what you can do have just become almost breathtaking. And I think the scientific community is a little bit sort of awkwardly positioned between the enormous power of the new tools they have available from the combination of stem cell research and organoid research and now human embryo model research. The capacity to build tiny little, livers and tiny little kidneys that you could use very easily to understand liver disease and kidney disease and all sorts of other basic cellular regenerative processes is so powerful. But the human embryo model, in a way, is an obvious culmination of that set of technologies, but it's happened fairly quickly. And it's happened for reasons that scientists didn't anticipate. It wasn't anticipated that you could take a skin cell and, make it turn into a liver cell. Like that just went against everything a biology textbook would have told you in 1990. But now that's just routine, and much more than that as well. So it's very understandable why it's not just embryo models, it's a whole range of other things too. the utility is vast. The scale of it is potentially vast. The diversity of the kinds of models you could make is potentially vast. The potential to industrialize it is huge. And of course, the possibility to commercialize it is huge. So all of these things are really big questions, which is why we think that this particular history about this particular issue of IVF and embryo research and how they were regulated has, in fact, much wider implications for biotranslational science now. And the Warnock consensus is actually something we should be thinking very carefully about in terms of what does it mean to have a social consensus about governance of very powerful scientific tools that do also have such wide range of applications. Yeah.
Thank you.
Actually, you should have said that question was from the chair of the Oxford International Biometric Centre. That organisation that
Okay.
it's an organisation Alabama was an active member of, and the organisation was actually initiated this annual series of events and on that. So thank you.
Oh, thank you. It's a subject that's very close to my heart because I just become a grandmother to IVF twins. But I'm a non-scientist, so I'm going to ask a really stupid question, I'm afraid. What's the difference between an embryo model and an embryo? And what are the implications if that was implanted into a uterus and so on?
Well, like Emily said, no one knows exactly. But if you make an embryo model, let's say that's an amalgamation of different parts of embryos that are assembled together into an embryo that essentially looks, functions, and behaves like a normal, embryo, the assumption is, that certainly could continue to develop. And the assumption is that, you probably could implant it and it probably would, become some kind of entity, and that probably will be done, with animals. But you can see how the ethical implications of that are quite considerable. You can see how not just the ethical implications for the entities involved, but also for the wider society. What would it mean for something like that to be condoned, as opposed to responsibly regulated? Clearly, it would be better if it was responsibly regulated. But in order to do that, you're dealing with a situation that's so complicated. You are definitely going to have some hard and fast rules. And right now, that is mainly that they won't be used for reproductive purposes. That's the one kind of principle that seems to be more or less universally agreed. But that's not a legal principle anywhere. That is just purely a kind of agreed constraint within the scientific community.
Just one thing to add to that. I think the key thing with embryo models is that they vary. They're not all the same. So I think the Naomi Morris's Lego analogy, I think, is quite helpful. You can make an embryo model which looks fairly complete. You can also make one which is very much incomplete and has absolutely zero potential ever of becoming anything, because it's just a little bit of the embryo developing. So I think the fact that some of them, there's no potential at all. The more complete ones, and there aren't many labs that are making embryos that look complete. The more complete ones, there are just all these questions we just don't know about them. So there's a lot. There's huge things that we don't know. One of the other things, just though, to say in relation to the interaction between this research and reproduction is scientists are also in mice making sperm and eggs from stem cells. And in mice, they've been successfully used to create mice offspring. So there are all sorts of people, scientists, working at the moment on creating human sperm and human eggs from stem cells, which is not going to happen anytime soon in the UK, but it could happen in the not too distant future. And that could be transformative, because what that would mean is somebody who didn't have their own eggs or sperm, perhaps they've had treatment for cancer or infertile, would be able to create sperm or eggs, which would potentially be transformative for lots of people. So there's another aspect of a very similar scientific branch of science which could have real impact on people's reproductive rights. But embryo models won't. There's a consensus that this is not for reproductive purposes.
So I'm as I've been seeing kind of the rolling back of legislation involving IVF and abortion and this. Because we couldn't create legislation that gave us time that it was probably necessary. Do you see a path forward for countries? I guess every other country besides the UK to kind of create some sort of legislation that would allow for productive research?
I mean, there are other countries where this research is being done, so it's not as though the UK is the only place where innovative research is possible. And so there are countries with liberal regimes, but there are also countries where haphazardly the way in which their laws were drafted is having an inhibiting effect on research. So Australia is a good example of that, because what happens with law is you pass a law and then 20 years later, that law is still in place, but science has superseded it, and there's something that wasn't expected. And embryo models are a good example of that. And in the UK, the assumption is they're not regulated. In Australia, the drafting of a piece of legislation they have, which was intended to prohibit cloning, looks like it prohibits the creation of embryo models as well. So legislation can have all these haphazard effects because it's always lagging behind the law. I think the possibility of comprehensive legislation in the US is a lovely idea, but I think there are definitely some challenges there. I mean, after the Alabama ruling, which I think had us all reeling, along with other things. But yeah, I think it would be great, but I'm thinking that in the US, presumably the obstacles are just vast.
It is interesting, though, in relation to the Alabama ruling, which was a ruling that was attempting to create the so-called personhood of the embryo, which is a long-standing tactic of the pro-life movement to create personhood for the fetus, personhood for the embryos through all sorts of very unusual means. And one of the sources of public outcry was that it would disrupt IVF, and that all the couples who had frozen embryos in IVF clinics would, not only have their treatment stopped, but would potentially lose access to those. And I think there was a very strong public sympathy for those couples, and there was a lot of criticism of the Alabama ruling because it's essentially a religiously derived position, which is being imposed through what you could describe as somewhat underhanded and undemocratic means. But there was also a lot of criticism because of the direct effect on people having IVF. And I think IVF occupies a very interesting position, what you might say sociologically and culturally, because IVF is, technically not something that could be supported ever by someone who thinks that life begins at conception. If you have the moral position, that life begins at conception and that all embryos are ensouled and therefore precious and therefore deserving of protection, you can't have any IVF, you can't have any embryo research. But as we know, IVF has become quite a normal, familiar, accepted, and in many ways celebrated technique. And so to say life begins at conception and all IVF and embryo research is therefore immoral, or even, to be precise, evil, is a less acceptable position because of IVF. So there's an interesting kind of, as I say, sort of looping effect where IVF itself has begun to be a driver of changes in perceptions of the relationship between biology and technology. And that's, again, something that we would want to pay attention to, because what that means is that IVF has itself become a kind of symbol of the way in which a relationship between technological modification of life is a way of creating life, as it were.
Thank you both, that was wonderful. I've been monitoring the comments online and I just wanted to read one out from Emma Cave, who says, thanks for a fabulous talk and congratulations on the book. I've ordered it and I'm looking forward to reading it. And also just a question from me. So I've done some research about patient and public involvement in health research, and it was really fascinating to hear about Anne McLaren and Warnock's approach and attitude towards public consultation. I was wondering if you could say anything about how that might translate into research today, because there is a push from health research funders to do it, but I'm not sure if that is universally welcomed or done in a sort of holistic way.
Well, interestingly, Anne's daughter, who isn't here, Susan Mickey, has argued that the recent government handling of COVID would have really benefited from a much more robust engagement with the public, a much more honest expression of uncertainty about scientific advice, a much more transparent process of public debate over, you know, pros and cons of this and that. And she's among many people who, following in her mother's esteemed footsteps, you know, argues that making scientific uncertainty more available to the public doesn't decrease public confidence in science, it increases public confidence in science because, you know, it's not, as it were, rocket science to figure out that you're not going to know if the vaccine's going to work 100% of the time if you just invented it. Hello, you know, that's obvious. So it's trying to tell people that it isn't, you know, that, oh, we're 100% sure of everything, can have the opposite effect. It can make people doubt the authority, the legitimacy, the credibility, the honesty of what they're being told. And there's even a word for this, you know, uncertainty value. When people are told this might work, it might not work, we're not really sure, we think it will, we've done our best, but, you know, we'd have to test it to really know. And, you know, we'd like your help in making sure that we make it as good as it can be, is very different from saying to people, we're absolutely 100% sure that if you buy this product, you'll be satisfied. You know, they're very, very different messages. But to understand that, of course, you need a more sociological understanding of biological translation, which is true for health, definitely as well.
Yeah, I think Sarah and I were talking recently about how interesting it is that Mary Warnock and Anne McLaren had this attitude towards public engagement really so early in the 1980s, because most people trace the drive to engage the public to the response to GM foods. The huge backlash there was because GM foods were being introduced and people didn't know about it and people were mistrustful. And so I think there was a real change in the early 2000s following the GM foods thing to say we need to involve the public. But actually, Mary Warnock and Anne McLaren were doing it 20 years earlier. And I think as a result, the Human Fertilisation and Embryology Authority has consulted at various different times in all sorts of ways. I think it's become part of just almost its DNA that consultation is part of what it does. And I think that's healthy. I think it's, you know, I think this sort of interesting contrast, isn't there, with the slide that Sarah presented about the letters that the Warnock Commission had received, which were massively not in favour of it. So if you govern by opinion poll, you don't necessarily get anything very useful. But if you talk to people and you do the sort of public dialogue work that's been going on, public engagement, real genuine two-way dialogue, I think it's incredibly productive.
Yeah. Another question? No, if not, then Sarah and Emily are staying for a drink, so you can get a chance to ask questions then. And if you want copies of the book signed by the two authors, the book's on sale at the back. But before that, can you just join me in thanking the both speakers for an absolutely fantastic presentation.